So today we're going to review for the Unit 8 test, which is tomorrow. So a few things that you need to know about the test, just things you need to concentrate on as you're studying. There are 40 questions on the test. About 15 of them are about DNA and RNA structure. Things like, complementary bases and the components of the nucleotide and all that kind of stuff that was on the last test. And then the rest are about transcription and translation and mutations. So it's all about DNA, RNA, protein synthesis, all that kind of good stuff. And then there are 2 bonus questions worth 3 points each. They're multiple choice this time. Hopefully that makes it easier for you. But that's what's on the test, pretty much like usual tests. So transcription is the first step in protein synthesis. There's an enzyme called RNA polymerase that unzips a portion of the DNA molecule and then matches up complementary RNA nucleotides and attach them together and then zips the DNA back up. And this produces messenger RNA, which can take the DNA message out of the nucleus to a ribosome, so to the location where the proteins are made in the cell. Translation is changing languages from the language of bases to the language of amino acids. Remember, the transfer RNA is kind of like the translator. Okay, it occurs at the ribosomes in the cytoplasm. The process involves messenger RNA attaching to the ribosome. Remember, ribosomes are made of ribosomal RNA plus protein. Transfer RNA brings in the appropriate amino acids and puts them in place by matching their anticodon to the messenger RNA's codons. And then the amino acids are attached together by the ribosome to form the protein, forming a peptide bond, which is the bond between amino acids. When you're reading the code, remember that your codons are read in groups of 3 bases. There are 64 total codons for 20 amino acids. Most amino acids have 2 or more codons. Methionine has only 1 and is the start codon, AUG. Tryptophan has only 1. Its codon is UGG. You don't necessarily have to know that because you'll have a codon chart. And then there are 3 stop codons. Big thing to remember always, always, always when using the codon chart, you're always using the messenger RNA to determine the sequence of amino acids. If you need to know the transfer RNA, remember it's going to be complementary to the messenger RNA. Another big hint, the transfer RNA anticodons are going to be the same as the DNA codon that originally coded for the messenger RNA, except they'll have uracil instead of thymine. But you don't really necessarily have to know that. It just makes it convenient to check yourself to make sure you have the right thing down. But most of the time you're going to be asked about the codon on the messenger RNA. Anticodons are found in the transfer RNA. They're complementary to the messenger RNA. The big thing is each transfer RNA carries a particular amino acid on the end that's opposite its anticodon. And then once the transfer RNA releases its amino acid, it's then free to go pick up another amino acid of the same kind. And like I said a minute ago, transfer RNA anticodons are the same sequence as the original DNA with a U instead of a T. Just so you can check yourself. Mutations are changes in the DNA. Generally, when we talk about gene mutations, we're talking about point mutations. This is changes in one or a few nucleotides. The substitution substitutes one amino acid for one nucleotide for another. It may or may not change the protein. Sometimes it does, sometimes it doesn't, but it usually only changes one amino acid. And so if you had to choose which one was least harmful, that would probably be the one that would be least harmful. But knowing that you can just change one amino acid can change something entirely and cause a problem, doesn't mean that they're not ever harmful. They're just probably the least harmful of the group. Insertions and deletions are going to change everything from the point of the mutation onward. That's called a frameshift mutation. Basically, it changes the reading frame of the nucleotides in the sequence. It's going to change everything from that point forward. And there are different kinds of mutations as a result of that. Quick video about transcription.
What you are about to see is DNA's most extraordinary secret. How a simple code is turned into flesh and blood. It begins with a bundle of factors assembling at the start of a gene. gene is simply a length of DNA instructions stretching away to the left. The assembled factors trigger the first phase of the process, reading off the information that will be needed to make the protein. Everything is ready to roll. 3, 2, 1, go. The blue molecule racing along the DNA is reading the gene. It's unzipping the double helix and copying one of the 2 strands. The yellow chain snaking out of the top is a copy of the genetic message, and it's made of a close chemical cousin of DNA called RNA. The building blocks to make the RNA enter through an intake hole. They are matched to the DNA, letter by letter, to copy the A's, C's, T's, and G's of the gene. The only difference is that in the RNA copy, the letter T is replaced with a closely related building block known as U. You are watching this process called transcription in real time. It's happening right now in almost every cell in your body.
Okay, quick video about translation.
When the RNA copy is complete, it snakes out into the outer part of the cell. Then, in a dazzling display of choreography, all the components of a molecular machine lock together around the RNA to form a miniature factory called a ribosome. It translates the genetic information in the RNA into a string of amino acids that will become a protein. Special transfer molecules, the green triangles, bring each amino acid to the ribosome. The amino acids are the small red tips attached to the transfer molecules. There are different transfer molecules for each of the 20 amino acids. Each transfer molecule carries a 3-letter code that is matched with the RNA in the machine. Now we come to the heart of the process. Inside the ribosome, the RNA is pulled through like a tape. The code for each amino acid is read off, 3 letters at a time, and matched to 3 corresponding letters on the transfer molecules. When the right transfer molecule plugs in, the amino acid it carries is added to the growing protein chain. Again, you are watching this in real time. And after a few seconds, the assembled protein starts to emerge from the ribosome. Ribosomes can make any kind of protein. It just depends what genetic message you feed in on the RNA. In this case, the end product is hemoglobin. The cells in our bone marrow churn out 100 trillion molecules of it per second. And as a result, our muscles, brain, and all the vital organs in our body receive the oxygen they need.
And just in case you need a little review, here's the replication video.
Using computer animation based on molecular research, we are now able to see how DNA is actually copied in living cells. You are looking at an assembly line of amazing miniature biochemical machines that are pulling apart the DNA double helix and cranking out a copy of each strand. The DNA to be copied enters the production line from bottom left. The whirling blue molecular machine is called helicase. It spins the DNA as fast as a jet engine as it unwinds the double helix into 2 strands. One strand is copied continuously and can be seen spooling off to the right. Things are not so simple for the other strand because it must be copied backwards. It is drawn out repeatedly in loops and copied one section at a time. The end result is 2 new DNA molecules.
So study for your test.